Have you ever wondered why some genetic traits remain unchanged for generations, while others slowly disappear? And how can scientists tell whether evolution is happening without waiting thousands of years? Today, we'll explore one of population genetics' most powerful concepts, Hardy-Weinberg Equilibrium. Don't worry if the name sounds difficult. By the end of this video, it will be simple. Hardy-Weinberg Equilibrium provides a mathematical model to predict genotype frequencies in an ideal population using allele frequencies. Its famous equation is p squared plus 2pq plus q squared equals 1, where p and q represent allele frequencies, and together, p plus q equals 1. Here, p squared represents homozygous dominant individuals, 2pq represents heterozygous individuals, and q squared represents homozygous recessive individuals. But this equation is more than just math. It acts as a null hypothesis, a genetic baseline showing what happens when no evolutionary forces disturb a population. If observed frequencies match the predicted frequencies, the population is in equilibrium. But if they don't, that means mutation, selection, migration, or genetic drift may be at work. And that tells us one fascinating thing. Evolution is happening. So in this video, we'll break down this concept step by step and make population genetics easy to understand. All right, everyone. Imagine we are looking at an entire population of butterflies. Some butterflies carry the allele capital A, and some carry the allele small a. Now, here's my question. If I know how common these alleles are, can I predict how many butterflies will be 2 capital A small a, or 2 small a? Believe it or not, yes! And that's exactly what the Hardy-Weinberg Principle allows us to do. Let's assume the frequency of allele capital A is 0.6, and the frequency of allele small a is 0.4. Notice something? 0.6 plus 0.4 equals 1, because these are the only 2 alleles in this gene. Now let's build a Punnett square. One parent can contribute either capital A or small a. The other parent can also contribute capital A or small a. Every box tells us the probability of a genotype. First, capital A meets capital A. That gives us 2 capital A. Its frequency is 0.6 times 0.6 equals 0.36, or 36% of the population. Now, capital A meets small a. That gives us 1 capital and 1 small a. 0.6 times 0.4 equals 0.24. But wait, the father can give capital A and mother can give small a? Or mother can give capital A and father can give small a? That happens twice. So 0.24 plus 0.24 equals 0.48, or 48%. Finally, small a meets small a. 0.4 times 0.4 equals 0.16, or 16%. Now let's check. 36% plus 48% plus 16% equals 100%. Perfect. This is why we write p squared plus 2pq plus q squared equals 1, where p squared is 2 capital A, 2pq is capital A small a, and q squared is 2 small a. Here's something students often misunderstand. P and q do not represent genotypes. They represent allele frequencies. The genotypes come after combining alleles. Remember this small difference. It can save you marks in exams. But here's the interesting question. Can every population in nature follow this equation forever? The answer is only if 5 important conditions are satisfied. 5 assumptions of Hardy-Weinberg equilibrium. Imagine nature is a perfectly balanced classroom. Nothing changes, nobody cheats, nobody leaves, nobody joins. Everything stays exactly the same. Only then can Hardy-Weinberg equilibrium exist. Let's see those 5 conditions. Condition 1, large population. Imagine only 10 rabbits live in a forest. If just one rabbit dies, allele frequencies change dramatically. But imagine 10 million rabbits. Would one death matter? Not really. That's why we need a large population. Large populations prevent genetic drift, which is random changes due to chance. Condition 2, random mating. Now imagine, every tall person marries only tall people, or every white flower pollinates only white flowers. Would allele frequencies stay random? No! Therefore, individuals must mate randomly, without choosing partners based on genotype. Condition 3, no mutations. Mutations create new alleles. Imagine suddenly an allele capital A changes into small a. Now the frequencies change immediately. So, for Hardy-Weinberg, we assume no mutations occur. Condition 5, no natural selection. Imagine red beetles survive better than green beetles. Generation after generation, red beetles become more common. Nature has selected them. Allele frequencies change, but Hardy-Weinberg assumes every genotype survives and reproduces equally. No one has an advantage. Final emotional ending. Now, look carefully. These 5 assumptions almost never happen together in nature. Nature is constantly changing. Animals migrate, mutations occur, natural selection acts, random events happen, people choose mates. Which means real populations are almost always evolving. That is why Hardy-Weinberg equilibrium is so powerful. It gives us a baseline. Like measuring a person's normal body temperature before checking for fever. If a population deviates from Hardy-Weinberg expectations, we know something is driving evolution. And that is the beauty of this principle. It doesn't just tell us when nothing is happening, it helps us detect when evolution is happening. Remember this simple rule. Hardy-Weinberg doesn't explain evolution, it helps us detect it. If even one of the 5 assumptions is broken, evolution has begun. That's the key idea you should never forget. Now that we understand the concept, it's time to look at some examples. I'll solve them step by step so you can follow the process easily. Pause the video whenever you want to try solving on your own and replay the solution as many times as you need until it becomes clear. The more you practice, the easier Hardy-Weinberg calculations will feel. So let's start with our first example. Before moving ahead, let's look at some common mistakes students make while solving Hardy-Weinberg problems. Mistake number 1, using phenotype frequency directly as Q². Remember, only the recessive phenotype directly gives you the value of Q². Mistake number 2, forgetting that 2pq represents all heterozygous individuals, not just disease carriers. Mistake number 3, assuming Hardy-Weinberg equilibrium without checking its 5 assumptions. Always verify the conditions first. Mistake number 4, using percentages instead of decimals. Always convert values properly. For example, 25% becomes 0.25 before calculation. And here's a pro tip. If you were given the frequency of the dominant phenotype, remember this. Dominant phenotype equals p² plus 2pq. So first, calculate q² by subtracting that value from 1 and then continue your solution. Avoid these mistakes, and your calculations will become much more accurate. Before we finish, let's do a quick revision of the most important Hardy-Weinberg formulas. If you know the allele frequencies, P and Q, you can calculate genotype frequencies using P squared, 2pq, and Q squared. If you know the recessive phenotype frequency, you can find allele frequency using Q equals the square root of recessive frequency. If you know the dominant phenotype frequency, first find Q squared by subtracting it from 1, then calculate Q. And if you were given the number of individuals with a genotype, simply use frequency times total population to find the expected number. To check whether evolution is happening, compare the observed values with the expected values. And most importantly, memorize these two golden equations. P plus Q equals 1, and P squared plus 2pq plus Q squared equals 1. Remember these, and you can solve almost 90% of Hardy-Weinberg problems with confidence. Before we end, here are some key takeaways that will help you remember Hardy-Weinberg Equilibrium for a long time. I hope this lesson made population genetics much easier and more interesting for you. If this video helped you, please like, share, and subscribe to support the channel and stay connected for more easy-to-understand biology lessons. Thanks for watching, and I'll see you in the next video.